So, good morning. It's 6:10 a.m., I believe, in California. This set of slides originally came from Tim Parrott, and I think he actually presented it at a ChemAxon UGM. And I recycled and presented it at a KNIME UGM, and now I'm recycling again and presenting at a ChemAxon UGM. But Tim was more of a scientist. He's more of a scientist, more interested in the users. I'm a techie, and so I'll do techie stuff. So DART NeuroScience, one slide. Our goal is quite substantial to try to get small molecules to maintain cognitive vitality, long-term memory. Not even a real indication. No clinical endpoint, not an easy job. Currently about 200 FTEs, or it's like 240 now. With the build-out expected about 260. We got a new building I'll show on the slide. And it's a privately held LLC by a single individual who owns a couple other companies. Scientific computing is my department, and I'm very proud that I've hired a lot of scientists and basically turned them to the dark side, turned them into informaticians and software coders. And so we don't act as a service. We try to collaborate as scientists, and so we know what they're doing. We know we've done it before. It makes everything flow pretty smoothly. The scientific computing group also, I think I got that next slide. Yeah, so this is the scientific computing group. We have computational chemistry. The cool part is basically that the information management and the software development is all hooked together with the project support. So it's all under one ladder. And so we don't get this handoff between informatics and comp chem. So the comp chem group does mostly standard comp chem modeling, structure-based drug design, library design. They're in the pre-LO space, LO and preclinical candidate space. I'm just reading these now. Project support for bioinformatics is more around target ID, target validation, that kind of stuff, expression analysis pathways. We have an open position. I put little flyers out there. If anybody wants to be a bioinformatician and live in San Diego, there's a slot. There's also a slot in our information management group. I have a very, very good team. My team leader had to leave because the commute didn't work, because we had that new building, she couldn't get to it. And then I have a dedicated group of software engineers, which is a really nice thing to have. These guys are very talented. And there again, I'm looking for one more software developer. But Alex said I can't have anybody from ChemAxon, so don't touch that. The advertisements are out there, but if Alex sees you and you're from ChemAxon, just walk away real quick. So this is Tim's slides. 200 plus scientists, 50 chemists, and we're doing parallel synthesis. So I don't know why, but we're doing parallel synthesis, and so we need a design tool. Tim's slide, this is the cycle you use when you do Combi-Chem. So the goals, we've constrained, you don't want to beat up your IT and IM. You're trying to move as much as you can out to your chemists and to get them more involved with the process. This is the way you scale, right? So I was at Pfizer for years, so I got good at trying to make things work for med chemists so that the med chemists themselves can do their own, play with their own stuff. The approach is very SOA. And so I could just, everything James said, just remember all that. For visualization analytics, Spotfire is our tool of choice. For chemical properties, calculations, reaction, enumeration, that's you guys, ChemAxon. We do a lot of 3D stuff. We use OpenEye there, and then KNIME is our tool of choice for data pipelining because of, as Serge said, pipeline pilot's ridiculously expensive for a small company. So the way, this is my slide. It's a geeky slide. It's, we're heavily invested in SOA. It's something I learned at Pfizer, and it turned out quite well. To me, I break SOA into two pieces. A CRUD kind of SOA, which is basically create, read, update, delete. So for an entity, you write a CRUD app. That's how you keep track of your entities. We have nice patterns relying on Backbone and Node and Twitter. And then we have the traditional stateless computational services. So there's the property calculations, enumerations, et cetera, et cetera. Services can be based on scripting. So you can write a C shell script, you can write a Perl script, Python script, doesn't matter. Services can also be written in NIME and run on the server. So basically what we're doing with the SOA is, and I'm just repeating what you've heard for the last 40 minutes, is move all the heavy lifting off into the server. So we did want our chemists to use this, and NIME is built off, you all know this, Eclipse, and Eclipse is a developer tool, and it's made for developers. It's not really that super friendly for medicinal chemists. Sorry, Aaron. So what we ended up doing is basically writing custom nodes that were named in what you would say the domain-specific languages, right? So the chemist looks at this and they go, oh, I know what a bioreactor and a tri-reactor and a unit reactor is. So I know how to batch details and how to do deduplication. We hooked it up to Spotfire, and then we have this very nice configuration panel that we've extended into NIME, which I'll explain why. Because what happens if you select a node in NIME, you usually have to kind of right-click and dig down and find the menu options. So what we did was we added a panel to the right that automatically gets populated when you click on the node. So it's nice for the chemists. So back to importing reactants, we keep them in a database. They're all highly curated. Chemists can choose the reaction type here. They're aldehydes, reactant types. Here's aldehydes. They can also use reagent number lookups, again, using the same services. So here we run deduplication. So this is actually a stateless service. So this one actually pushes the molecules to the server, runs the deduplication using ChemAxon, sends results back into NIME, and you can see what it does. When you have reactants that they have different salts, you don't want those all going into your combinatorial chemistry. They're just going to give you the same molecule, and so it deduplicates by salt stripping. This again shows the nice configuration panel. You just click on the bioreactor node, and then you get to see all the biomolecular reactions, complete with little pictures, right? So chemists can just scroll really quickly and find, and they're all highly curated. We take what ChemAxon provides, but then we tweak it to make it work in our environment. And here's an example, again, of a comp service. This is one where it's NIME on the server side. So the chemist sees a unireactor node, and really what's going on underneath is all this, try to point, all this stuff is going on the server. And this is the number of box removed, because you can't get all the box off. You have to keep looping, as I mentioned yesterday. Again, this is a stateless service. We call out to NIME, or we call from NIME out to our server, runs calculated properties, sends them back into the data pipeline. Same story again, clustering. So the chemists, all they see is a cluster node with a couple of simple parameters to choose from in that inspector panel, and then all of it's done server side with a NIME protocol. So we can do NIME heavy lifting on the back end again. Clustering is too much to do on our CombiChem people are designing up to 200,000 compound libraries, and so that's why we have to do our enumerations through server side. It's quite expensive. And this one's even more expensive. So we use ROCS as basically our similarity search, right? So basically to find out if it's based on known hits for the given target, they choose, well, I guess they choose ROFL1B, 3D queries, and they run it, and then it gives them a ROCS score for their library to see if they think it'll actually hit the target of interest. Oh, I missed this slide. And then it goes out of the ROCS calculations into Spotfire or Vita, so you can look at it in 3D. We had to do something because, like I said, we're running really long calculations with the enumeration, and the ROCS is even longer. So we actually had to add this thing to, this is something that was missing from pipeline, or this in pipeline pilot that's missing from NIME, is the ability to shut down NIME and come back later and have your protocol run. So NIME, when you turn it off, dies. So they would have to leave their computers open all night if they wanted to do a calculation. And so we had to extend KNIME so it has the ability to remember a job that it's launched and then pull it back in when it reopens. What is this one? Okay, then you can just go ahead and export it to Spotfire. So what happens is you export a very large library, maybe 200,000 compounds, maybe more, and you filter in property space, all the properties come along, and then you have to go back and calculate the reactants. We do full combinatorial matrices. So they filter in product space, and then they get reactants back out that they want to put back into KNIME. And so you export, you do your selection then in Spotfire. And then what happens, in Spotfire, we put a little thing that pushes it back into KNIME in these kind of, we call them rabbit nodes that kind of pop up that have the products and then the various reactants that were actually chosen in the Spotfire session. And so then you can continue on. We run a few more computational services to get the data we need. This actually generates what we call a library design plan. And the chemist doesn't actually see this. It actually just goes right into the electronic notebook. And so the library design plan comes out of KNIME and goes in. This is the Agilent ELN that Francois talked about yesterday. So that made a nice workflow. And the chemists like it. They use it, and we produced 90,000 compounds a year. So this was done fairly quickly. I have a very good team. Started, the chemistry group was in 2011, and then I guess by 2013 we had pretty much everything working, and it's been working in production since. One of our major developers, Aaron knows him, he's up in San Francisco. A lot of people may know him. Luty DeQueller, he's a very talented software engineer. Ron runs the methods group. Karen is a contractor. Kenny's a contractor. Zach's a contractor, and Daniel, and they've all been involved in the services and deployment. Testing is Eileen from information management, Andrew Burt, who runs the structure guided chemistry group, and then I do management with Melanie and Heather and PM. And that is all I have.
Thank you for your presentation. We have something similar in our company, which is good.
I think everybody has. If we just reinvent everything.
I would like to ask you, this library design, did you include the subset selection? Because sometimes, obviously, the combinatorial library will give you 50,000, but the chemists, they want to do 5 R1s and 10 R2. So, yeah. For example, you can include multi-objective genetic algorithm.
So you're
it was published by GSK.
Oh, you're talking about building a model to try to evaluate the libraries?
To make small libraries, because the one that you presented, the bioreactor, probably will do the whole combinatorial library.
Yeah. So I'm sorry, I didn't explain well, because you go into Spotfire and you make a selection. You look around in product space. You can look at the molecular weight of your product. You can look at C-log P of your product space and select in product space, and then back calculate the reactants that are needed. Does that make sense?
No, makes sense.
Then the reactants go into the KNIME, and you can do multi-step reactions, right, because it's KNIME. You just chain it together. So this is why it made sense to me to do CombiChem in KNIME, because basically you let people chain together multiple steps in the reaction. Last step, you put a debug node in there, and then it works.
What's your experience in terms of results when using ROCS and 3D similarity compared to traditional fingerprint-based similarity?
It destroys it. ROCS is so much better. It destroys it. We've tried fingerprints. We use fast ROCS. It gives us enrichments like 25-fold at 0.01% if we order from molecules.
Have you done any comparison of ROCS versus the Crescit Blaze tool, the Z field?
I've been asking some of my folks to try that for a while, but no, we haven't done a head-to-head, and I'd love to, because we do have the Crescit tool.
Okay.
Are you happy with the Uni B and 3 reactor nodes in KNIME, or would you prefer something like one reactor node which can decide?
No, because the number of ports change. So you don't want to dynamically, if you have a bioreactor, you have 2 input ports. If you have a trimolecular reactor, you have 3 input ports. If you have a unireactor, you have 1 input port.
Yeah, I know that. In fact, I was just asking that.
And it also helps segregate the data, right? A chemist knows a unireactor is usually a deprotection, right? They know bimolecular is more traditional, so most of the reactions are bimolecular, and then trimolecular is very rare, so it also categorizes.
Yeah, of course, but in this case, you have to have the right reactants for the first input and for the second input.
We automatically figure that out.
Okay, but we could do this that it can decide automatically. Would it be better or not?
No, I don't think so.
Okay, thanks.
They're already used to this. It's only 3 nodes. They can figure it out.
Okay.